Hi, my name is Julia Wilsey,
And I'm Madison Layer.
and we are in Dr. Dave Zanata's lab at Central Michigan University. Today we're going to be looking at 4 different studies from our lab that use geometric morphometric analyses and DNA barcoding to distinguish among various Great Lakes freshwater mussel species.
We're going to start off with a little bit of freshwater mussel history. For the past 200 years, mussels have been primarily identified by their shell characteristics. Due to a force called phenotypic plasticity, the shell shape of mussels within the same species can vary based on the water conditions and habitat it resides in. Small differences in shell morphology make it hard to determine if a particular mussel is a different species or not. This problem has been ongoing in the malacological field, including the species we are going to be looking at today, and has important implications for mussel species status assessments.
The species that we are going to be focused on in this presentation are Transilla truncata and Transilla dunassiformis, Fusconea flava and Plerobemus syntaxia, or the pigtoes, Pyganodon grandis and Pyganodon lacustris, or the floaters, and Lampsillus silicoidea, Lampsillus fasciola, Lampsillus cardium, and Ortmanniana ligamentina, which we are referring to as the lampsillines.
The objectives for our studies were to 1. Determine how well morphometric models discriminate between and among similar species, 2. Establish identifications of specimens using DNA barcoding, and 3. To determine the accuracy of field identifications compared to the confirmed species identities.
For all 4 studies, the first thing we did was collect specimens. Specimens were collected from various rivers in Michigan and Ontario, Canada. After the specimens were collected, an initial field identification was made. Specimens were measured, including the length, width, height, and hinge length of the mussels, and genetic samples were collected in the form of swab or mantle clips. At least 20 genetic samples were collected at each site, and swabs were stored in lysis buffer, while clips were stored in 95% ethanol. Photographs were then taken. When taking the photos, we took great care in making sure that the shell was laying very flat because we used these photos for geometric morphometrics later. After processing the samples, specimens were then returned to the river where they were found.
After completing the field portion of our methods, it was time to begin barcoding the specimens. We first extracted the DNA using either a QIAGEN blood and tissue kit for the mantle swabs or an isohelix buccal swab extraction kit for the swab samples. We then ran a gel electrophoresis to check for the quality of DNA. Afterwards, we amplified the female lineage CO1 region of the DNA using a polymerase chain reaction. A gel electrophoresis was run to check for proper amplification of the CO1 barcode. The next step was to purify the DNA, which we did using a solution of Exosap. Lastly, we had the Amplicon Sanger sequence at the Eton Bioscience Lab. After the samples were sequenced, samples with 400 continuous base pairs and a quality score over 20 were compared to CO1 sequences on GenBank. Sequences that were most similar were chosen as the most likely species for each specimen.
We then analyzed our specimens using geometric morphometrics. To do this, we have our specimen here from the field. Notice that the shell image is very flat. We place type 1 landmarks, or homologous points that are stable, at the tip of the umbo and at the end of the hinge ligament, except in the case of the pyganodons, where we put points on both ends of the umbo. Using the 2 points, a 40-ray fan was created. Type 2 landmarks, which are anchored by type 1 landmarks, were then placed at every point where the fan met the edge of the shell. From this data, a canonical variance analysis, or CVA, was used to determine if there were significant differences in shell shape between the study species. We were also able to create a Procrustes deformation grid using this data. We're firstly going to talk about the Transilla project. For this project, 103 CO1 sequences were amplified, including 68 Transilla truncata and 35 Transilla dunassiformis. The dataset also included 86 museum specimens that were compared to the LDA model later. The landmark dataset correctly assigned 99% of the specimens to their genetically confirmed species identities. You can see in the CVA that Transilla truncata are on the right in red, and Transilla dunassiformis is on the left in blue, with a very distinguishing line between the 2 species. If you look at the Procrustes deformation grid, the mean shape of Transilla dunassiformis is represented as the circles, and Transilla truncata is represented as the ends of the vectors. Based on the grid, Transilla truncata are more triangular in shape, while Transilla dunassiformis is more elongate. Moving on to the pigtoes, the results of the sequencing revealed that we had 133 CO1 sequences, 90 of which were Fusconea flava and 43 were Plerobemus syntaxia. Only 82% of specimens were accurately identified in the field, with misidentifications more common for Fusconea flava than Plerobemus syntaxia. Geometric morphometrics correctly assign 99.2% of specimens to their genetically confirmed species identities. As you can see in the CVA, Fusconea flava are on the right in the triangles, and Plerobemus intoxia are on the left in the circles. They also have corresponding foot color, either orange or white. You can see that there is a distinguishing line between the two species, and that both species have both orange and white feet. We also have our Procrustes deformation grid that shows Plerobemus intoxia in the circles and Fusconea flava in the arrows. You can see that Fusconea flava have more centrally located beaks than Plerobemus intoxia, and Fusconea flava are less round due to the sulcus on their shell. Based on this study, it was determined that landmark-based geometric morphometric analysis was over 99% accurate when compared to DNA barcoding results, and that geometric morphometrics is a useful tool in differentiating between these species. Foot color, however, was only 77% accurate in differentiating between the species and is much less accurate. In addition, imperiled Plerobemus intoxia may be more common than originally thought because they are often misidentified as Fusconea flava.
For the floaters, CO1 sequences were amplified for 212 specimens from Font Lake and Lake Gennesareth on Beaver Island, Michigan. These sequences resulted in 128 Pyganodon grandis and 62 Pyganodon lacustris. There were also 146 heteroplasmic specimens and 22 putative hybrids where the F and M-type mitochondrial DNA CO1 sequences did not match the species ID. The landmark dataset correctly assigned 97.4% of the non-hybrid specimens to their genetically confirmed species and lake of origin, while 90.9% of hybrids were assigned to the species ID of their F-type lineage. Axis 1 differentiated between Pyganodon grandis and Pyganodon lacustris. Axis 2 differentiated between Pyganodon grandis from Lake Gennesareth and Pyganodon grandis from Font Lake. Axis 3, not shown in this figure, differentiated between Pyganodon lacustris from the two lakes. Hybrid individuals correspond to their F lineage CO1 sequence, and they tended to span a larger area on the LDA. In the Procrustes deformation grid, circles represented Pyganodon grandis and arrows represent Pyganodon lacustris. Based on the grid, Pyganodon lacustris is shorter and more elongate than Pyganodon grandis. 15.1% of heteroplasmic specimens were hybrids, and further assessment of hybridization with microsatellites or SNPs is needed to look at first-generation hybrids. Based on this study, morphometric analyses are useful in confirming species identifications of the floaters. Moving on to the Lampsaline study, 248 CO1 sequences were amplified from specimens from 8 different rivers in Michigan and Ontario. Results from the amplification revealed 41 Lampsilus fasciola from 1 river, 96 Lampsilus cardium from 6 rivers, 28 Ortmanniana ligamentina from 4 rivers, and 83 Lampsilus silicoidea from 5 rivers. An additional 141 specimens were photographed for use in the linear discriminant analysis. This is the LDA that was created with the Lampsaline data. There are 7 groups representing the species and sexes. The solid points on the figure represent specimens with CO1 confirmed identification, and the open points represent the specimens with morphological data only. Axis 1 of the LDA accounts for 44.68% of the variation, and Axis 2 accounts for 31.2% of the variation. The specimens of each species are arranged in clear groupings, but there is some overlap, specifically between the female Lampsilus silicoidea and Lampsilus cardium. The Lampsaline LDA was found to be 85.5% accurate in assigning specimens to the correct group. This chart shows the comparison between genetic assignments from the CO1 barcoding and the LDA assignments. The bolded numbers show the specimens that were correctly assigned in the LDA. Accuracy within the LDA differed. For example, the highest accuracy was for male Lampsilus silicoidea, which was 96.6% accurate, and on the lower end, the LDA was only 76.2% accurate for the Lampsilus cardium females. Based on the results of all 4 studies, landmark-based geometric morphometric models in combination with DNA barcoding are useful in differentiating among morphologically similar mussel species, and our models can be useful tools for field biologists. These datasets are now available to be used in future studies.
We would like to thank our advisor, Dr. Dave Zanata, for helping us with every step of these projects, Fisheries and Oceans Canada, especially our co-author Dr. Todd Morris, and the CMU Office of Research and Graduate Studies for funding. We would also like to thank everybody who helped in the collection of specimens and the lab work for these projects. Thank you so much!